viewers. So we are studying PDH complex, and in the last video we have covered the 3 catalytic enzymes of PDH complex, and today we will explore about the 2 regulatory enzymes of this complex. As I told you, it overall contains 5 enzymes, 3 for catalysis, 2 for regulation. So today it's turn to study about regulation of PDH complex. So the 2 enzymes required for regulation of this complex are kinase and phosphatase, and their name is Pyruvate Dehydrogenase kinase and Pyruvate Dehydrogenase phosphatase. So kinases, wherever you find, their role is to do phosphorylation, or you can say covalent modification of enzyme complex, and phosphorylation means to add on phosphate, correct? And phosphatase do the reverse of kinase, means dephosphorylation or removal of phosphate. So the most committed enzyme of the overall cellular respiration is regulated by just 2 enzymes, the kinases and the phosphatases. Okay. So the point to be noted here is that both these enzymes are bound to the E2 subunit of the PDH complex. Okay. So let's explore a bit more about the detailed role of pyruvate dehydrogenase complex. What it causes, yes, it bounds to the E2 subunit of the PDH complex, but it causes phosphorylation of the even subunit which contains serine at the active site. Enzyme E1 of the PDH complex contains serine at the active site, and serine, the amino acid, it has OH as a R group. If you remember, we have discussed in the amino acid lecture much before. So kinases, wherever work, they use the ATP as a phosphate group donor and causes phosphorylation, converting the ATP to ADP, means they snatch a phosphate group from ATP. Here ATP act as a donor of phosphate group. So the kinases which are present on the E2 subunit of PDH complex causes phosphorylation of the even subunit. Clear? So this phosphorylation of PDH kinase on E1 switches off the activity of this complex. Got it? Means this enzyme is responsible for turning off the enzyme. Got it? And we have previously discussed that not all enzymes needs phosphate group for activation, and I have explained this enzyme with all of you in the previous lecture, and I would like that if you write the answer in the comment section. Okay. So let's move forward. So if there is an enzyme, there is an activator, there is an inhibitor. Okay. So in the cellular metabolism, there are various substrates, there are various products, and there are various enzymes, and all these are activated and inhibited at some time. You need not to activate every enzyme at every time. Okay. So the activator of this kinase is high levels of NADH, acetyl coenzyme A, and ATP. How these activate the kinase? See, NADH competes with NAD plus for the binding to E3. Remember the previous lecture where I told the mechanism of the E1, E2 and E3 complex together, where the very detailed picture I have given. In that, NADH comes and NAD plus comes and binds to E3, and it snatches protons from the FADH2, which is already present on the E3 complex. So what does NADH do? It competes with NAD plus for the binding to E3. That's why it causes inhibition of this complex. Second, acetyl coenzyme A. What does it do? It resembles with coenzyme A. So it competes with coenzyme A for binding to E2. Okay, and I hope if any of you has not watched the previous video, please watch that video to understand clearly this regulation. Okay. Now coming to the inhibitor of this complex. So what are the inhibitors of this complex? These are ADP and pyruvate. Pyruvate, which is a substrate of the reaction catalyzed by PDHC, is an inhibitor of this complex. ADP, what does ADP show? ADP show there is less energy. Okay. So coming towards the next slide, sorry. Yes. Now coming towards the next enzyme, which is phosphatase. So obviously E1 was phosphorylated by kinase. So what does phosphatase do? It causes dephosphorylation, and where it does dephosphorylation? Obviously where the kinase has put phosphate, and we know that kinase has put phosphate on the E1 subunit. So basically phosphatase causes removal of phosphatase by a hydrolysis reaction. You can see from this reaction. So dephosphorylation by pyruvate dehydrogenase phosphatase restores the activity of the complex, means it switches on the complex, or you can say when pyruvate dehydrogenase complex is active, means pyruvate dehydrogenase phosphatase is active. Okay, because it is the subunit which represents pyruvate dehydrogenase complex. Kinase does the opposite role. So coming to the activator of this complex, when you need this complex to be activated, or when you need the phosphatase to be activated, obviously when if their energy is low, means ATP is not there, rather ADP is present, and magnesium ion, calcium ion, insulin, pyruvate, NAD plus and coenzyme SH. Let's take one by one. ADP, you understand that if ADP is low, this phosphatase must be in activated form, because we need to synthesize acetyl coenzyme A from pyruvate, because we need to go to citric acid cycle to generate NAD, HS and GTP, and further to the oxidative phosphorylation to generate ATP. Why magnesium ion is an activator of this complex? I am going to explain this, and please be very careful. If you see the structure of ATP, the adenosine triphosphate, you will find that the phosphate groups or the negative charge of the phosphate groups are stabilized by magnesium ion, or in many of the reactions you will find that magnesium ion is also written on the reaction arrow along with ATP. So it means that magnesium ion stabilizes ATP, means if magnesium ion is much more in the cell, it means it is not bound to ATP, and if it is not bound to ATP, it means ATP is not much there in the cell. So basically the high levels of magnesium ion represents that the energy level or the ATP level is low. Clear? Coming to the next ion, the calcium ion. I have not discussed GPCR or G protein coupled receptor right now, but if you have any idea about it, it is a second messenger. Calcium ion is a second messenger. So when there is low levels of glucose or low levels of energy, the level of calcium ion is high. So it says that the cell requires energy, and if cells needs energy, the PDH complex should be activated. Insulin, again, what does insulin do? It causes the conversion of glucose to glycogen. Okay. Pyruvate, if pyruvate is high, means you need to go to acetyl coenzyme A to generate energy, and simultaneously the NAD plus and coenzyme SH. All these are the activator of this complex, because all these say that we need energy. If there is much glucose, it means we need energy. If there is much pyruvate, we need to go to the formation of acetyl coenzyme A. Clear? So what does activated kinase say? The activated kinase say that the enzyme is switched off, because there is no need for formation of acetyl coenzyme A from pyruvate, and when it happens, it happens when the ATP level is high. When you have much ATP, you need not more ATP to be synthesized. That's why glycolysis decreases and PDH activity is inhibited. Got it? Note down this point in your mind that PDH activity inhibited means kinase is activated. Got it? So, when this happens, acetyl coenzyme A will influx from beta oxidation to citric acid cycle and glycolysis will slow down. While the activated phosphatase causes switching on of this enzyme complex, and it means that when there is low energy, PDH works, and if PDH works, phosphatase should be activated because it is the phosphatase which actually represents the enzyme. So, if it happens, glycolysis happens, correct? So, one more important point which is written at the bottom of the slide is that the Pdp deficiency, Pdp means Pyruvate Dehydrogenase phosphatase. It is found that when the phosphatase enzyme is deficient, we have lactic acidosis, means we have increased production of lactic acid. Why so? If you remember the reaction catalyzed by PDHC, which is the conversion of pyruvate to acetyl coenzyme A, which is done by PDHC, or you can say PDP or pyruvate dehydrogenase phosphatase, because it happens when phosphatase is activated, correct? So, if phosphatase is not activated or it is deficient, what will happen? Cell will not undergo formation of acetyl coenzyme A, and if you remember in the previous lecture that where I discussed the fate of pyruvate, I told you that in anaerobic condition, pyruvate can undergo formation of lactic acid. So, when anaerobic condition arise, when pyruvate cannot undergo formation of acetyl coenzyme A. So, if PDP is deficient, pyruvate cannot undergo formation of acetyl coenzyme A, but we need glycolysis to happen. For that we need NAD+ to regenerate, and for that we need lactic acid dehydrogenase to come and do this role. And as a byproduct of that reaction, I told you that lactic acid is formed in the cell. So, if PDP is deficient, lactic acid formation causes at a high rate, which causes lactic acidosis, and which will lead to progressive neurological and neuromuscular degeneration, and which is often associated with epilepsy, correct? Now, coming to the more very beautiful picture representation of this complex, showing the kinase having a serine OH group and a serine which is phosphorylated by the kinase, which is shown in the step 2 right here, and it is the point where phosphatase comes and causes hydrolysis of this phosphate to again converts the enzyme into its inactive form or in a kinase form. So, it is a very beautiful picture, and through this you can easily understand that if concentration of NADH is high, what will happen? If concentration of acetyl coenzyme A is high, what will happen? And vice versa, if there is low concentration of NADH and low concentration of acetyl coenzyme A, what will happen? So, it is a beautiful representation of enzyme complex along with the kinases and the phosphatases, correct? So, coming towards questions, we have not discussed questions since long. So, I have noted down a few for you. The first one is conversion of pyruvate to acetyl coenzyme A is known as. So, you need to go to the previous lecture and you will find that it is oxidative decarboxylation, the first step. Okay. Second, which of the following is not the regulator of pyruvate dehydrogenase? So, we have studied right now that calciamide, yes, it is a regulator. Acetyl coenzyme A, yes, it is also regulate. The level of ATP, yes, it also regulate, but we have not seen citrate to regulate this complex. So the correct option is option D, or citrate. Third is an easy question. We have just discussed that which of the following enzyme causes congenital lactic acidosis, and we have just discussed that it is pyruvate dehydrogenase. Okay. So if you remember or if you understand what I explained to all of you, you will be able to answer this homework question clearly. So please be careful and write the answer in the comment section. I'll wait for your answers. Keep revising and happy learning.